So hello and welcome everybody to our new RPTH research recap, where we dive a little bit deeper into the science that has been published in our favorite journal. So my name is Timo Vögtl and I work as a senior postdoctoral fellow in the lab of Dr. Nieswand, and I'm based at the University of Würzburg in Germany, and I'm also an editor at RPTH. My research focus are platelet receptors and their function in health and disease. And it's my pleasure to introduce today's speakers, which are Doctors Erin Norris and Pradeep Singh, which are both based at the Rockefeller University in New York and will present the latest publication which was published in RPTH. And Erin Norris earned her PhD in pharmacological sciences at the University of Pennsylvania and completed her postdoctoral training at the Rockefeller University. She has undertaken numerous lecturing and research roles, as well as scientific journal editing positions. So currently she's working as a research associate professor in the Laboratory of Neurobiology and Genetics at the Rockefeller Institute in New York. And so she's doing actually cutting edge research, and she's investigating the role of vascular dysfunctions in Alzheimer's disease pathophysiology. And she also develops novel biomarkers and therapeutics. And she also has a second research topic where she focuses on the role of the plasma contact system. And Pradeep Singh obtained his PhD at the IIT in Bombay in India, where he explored the toxic protein aggregation of alpha-synuclein in Parkinson's disease pathogenesis. And at the moment, he's working as a research associate in the Laboratory of Neurobiology and Genetics at the Rockefeller University in New York. And there he explores how vascular factors and the intrinsic coagulation pathway can exacerbate neurodegeneration, blood-brain barrier damage, and dementia in Alzheimer's disease. And that's also the topic of today's presentation, which is entitled Blocking Domain 6 of High Molecular Weight Kininogen to Understand Intrinsic Clotting Mechanisms. So the talk will be followed by a Q&A session. So for those joining us live on Facebook, Twitter, and YouTube, please leave your comments below, and then they can be answered during the Q&A session. And yeah, stage is yours. Looking forward to your presentation.
Thank you very much, Timo, for the nice introduction. And thank you to our viewers who are here that are interested in our work. And also a big thank you to RPTH for inviting us to present our publication. So as Timo said, we'll be discussing this recent publication. And I wanted to start off the discussion by introducing or reviewing the contact activation system. So our bodies have two ways of clotting our blood when it's necessary. And the most important way that the system that we cannot live without is called the extrinsic clotting pathway. So when we're injured, the extrinsic clotting pathway is what helps us stop bleeding. The other pathway that induces clotting is called the intrinsic clotting pathway. And this is a system that some organisms don't even have, but humans still have it. And this is part of the contact activation system. And this is introduced with coagulation factor 12, which we see here. And it's activated when it binds to a negatively charged surface, and it becomes factor 12A. Now, factor 12A then can activate coagulation factor 11 to factor 11A. And this sets off a series of enzymatic events, which eventually give us thrombin, which cleaves fibrinogen into fibrin, which is the major component of a blood clot. So that can give us then vessel occlusion and coagulation. Factor 12A can also set off an inflammatory pathway. And this occurs when factor 12A cleaves prekallikrein, or PK, into kallikrein, or PKA. And PKA is very important because it cleaves high molecular weight kininogen, or HK. And when it cleaves HK, it gives us a potent peptide, a potent inflammatory peptide called bradykinin. It's only made up of 9 amino acids. Another important thing about PKA is that it can feed back on this entire system. So it can reactivate the entire system so that it perpetuates itself. So the system can be, even though it's not a major system in our bodies, it can cause a lot of coagulation and it can cause a lot of inflammation when it's out of control. Another very important aspect of the contact activation system, as you can see circled here, is HK. So not only is HK directly responsible for producing or generating bradykinin, this potent inflammatory molecule, but it is also necessary to be bound with factor 11 or PK. And factor 11 and PK, when bound to HK and they circulate in the blood this way, that is how factor 11 and PK can become efficiently activated by factor 12A. Now, the system is very complicated, but when it's dysfunctional, it can be cause a lot of issues in different diseases and disorders. And this has been shown in numerous diseases that the contact system is dysfunctional. So we explore its activity and its dysfunction in Alzheimer's disease, but it's been shown by others that it's active in COVID-19, multiple sclerosis, lupus, sepsis, IBS and colitis, and hereditary angioedema, as well as many other disorders and diseases, as you see here. So we wanted to better understand how this pathway works and how it can be dysfunctional, and potentially be able to see if we can use a molecule or use some sort of system to stop dysfunction of the contact activation system. And we were able to do this by studying using a special tool that we have, using normal pooled plasma from humans, as well as factor deficient human plasma, and then looking at contact activation assays. We were able to kinetically explore clotting using a spectrophotometer. And now the tool that we used is an anti-HK antibody, which was generated in our lab. We refer to it as 3E8. And what is special about this antibody is that it binds to domain 6 of HK. And this is the region where PK and factor 11 bind to HK. So when you add 3E8 to blood, it will actually displace factor 11 and PK from the complex with HK. And in doing this, it also prevents HK from being cleaved by kallikrein, so we don't get bradykinin generation. And one other thing I want to point out about this is that when we use 3E8 in our assays, we used it at the same physiological concentrations of HK. So I'm just going to start off the talk with our work. When 3E8 is added to blood with HK, it displaces PK and factor 11. From HK. And also something that was very important to show, we can look at intrinsic clotting versus extrinsic clotting by, you can do an assay called APTT, which is activated partial thromboplastin time. And that specifically looks at intrinsic clotting. And it uses ellagic acid to stimulate the clotting pathway. And when you put normal plasma in this system and add ellagic acid, you have very quick clotting of the plasma. But when we add 3E8 to the system, it significantly delays how long the clotting mechanism. If we use IgG instead of 3E8, it has the same clotting time as normal control. Now in prothrombin time, which is where we look at extrinsic clotting that does not work through factor 12 and factor 11, 3E8 has no impact on it. So clotting occurs very quickly in the presence of 3E8. So 3E8 specifically works by intrinsic clotting mechanisms. Okay, so Pradeep now will take us away, and luckily his slides are here.
Thank you, Erin, and thank you, Timo, and thank you, RPTH, for giving us opportunity. So I'll just talk the next things that we know that 3E8 is blocking the PK and factor 11 binding to HK, and that's how it's delaying the intrinsic clotting. So the next idea was that if, as you can see here in the red, like the normal pool plasma, which is NPP, it's clotted here, but when 3E8 is there, it's significantly delayed. So the next idea was 38 is preventing the binding of HK, like HK and factor 11, which affects its activation. So whether we can add already activated factor 11 and like nullify the effect of 38. So I just added factor 11A, which was much less than the physiological concentration of factor 11, because in the disease or in the normal conditions, not all the factors get activated, like not 100% activation happens. So we just choose 1.5 nanomolar, which is roughly 5% of physiological concentration of factor 11. And you can see here, like there's a dramatic reduction in the clotting time, means the effect was 3.8 was nullified because you already adding the activated factor 11. And because I didn't mention that the PK is also binding to HK, and PK gets activated and feedbacks the pathway of intrinsic clotting. So, and 3.8 also blocks the PK binding to HK because they both have the same, like similar binding site. So the binding site of PK and factor 11 to HK overlaps. So that's why it blocks the binding of PK and factor 11 both. So you can see here, by adding the PKA, which is activated form of precalcitonin, you also see the correction in the clotting. So this is very clear that how 3.8 is working here. The next slide. So now we mentioned that this antibody is specific for 3.8, it's specific for HK. So when we use the HK deficient plasma, which ideally did not have any HK, like high molecular weight kininogen, the effect of 3.8 was not seen. As you can see here, the normal pool plasma, which clots much faster than the HK deficient plasma, which is expected because HK has a role in the intrinsic pathway, and 3.8 does not have any effect. But what is interesting here, you can see like HK deficiency, still the plasma is clotting, even though it's delayed. And more surprisingly, the HK deficient individuals, they don't have any bleeding complications. So they are usually normal. They don't have any bleeding complication like factor 10 deficiency or factor 9 deficiency. So then the question is that how this HK independent pathway is working in the physiological condition, like in the body. So when you add HK to HK deficient plasma, you'll see that nice correction of clotting, which is expected because you are adding back the purified HK. But when the HK was pre-incubated with 3.8, so it binds to the domain 6, you don't see that much correction because 3.8 is bound to the added HK, and now the factor 11 or PK in the blood cannot come and bind to it. So that means this is how further we explain how 3.8 works. And the next thing what I did, because HK is the only non-enzymatic component, it does not have any enzymatic like activity. It only helps PK and factor 11 for activation through intrinsic pathway. So then my thing was that whether we can add already activated factor 11 or PKA and see what happens in the HK deficient plasma. So you can see here when you already add the factor 11 deficient, like factor 11 purified factor 11A and PKA, the clotting delay was almost nullified. Like it's clotting like similar to the normal pool plasma. So that gives an idea that in the case of HK deficiency, if there is a sufficient factor 11 is activated, and now recently it has been proposed that factor 11 can get activated by thrombin, so then you don't need the HK. And that could be the reason why HK deficient individual are not showing any bleeding complication from our study. That concludes. The next slide. And one interesting, sorry, the slide had some problem. It doesn't show the title. But what I want to highlight here, this antibody is very peculiar in the case that it blocks the binding site of both PK and factor 11 to HK because they have the similar binding region. So in the normal pool plasma, as you can see here left, either PK binds to HK domain 6 or factor 11 binds. But when 3.8 is there, as you see in the middle, it displaces or it doesn't allow PK or factor 11 to bind. So now there are plasma available, the human plasma, like which is PK deficient and factor 11 deficient, the single deficient plasma. So using that plasma, we can further explore the clotting pathway using the 3E8 as a tool. The next slide. Sorry for this uh, title thing. So, but I just want to highlight here when we use the PK deficient plasma, which only has factor 11, it does not have PK. So only 3A blocks the factor 11 binding. And as you can see here in the PK deficient plasma, which is only slightly uh, delayed compared to the normal pool plasma clotting time, the effect of 3A was very dramatic because it blocks the factor 11 binding to the 3A, factor 11 binding to the HK. And as expected, when we add activated factor 11 or PKA, it corrected the clotting delay. And so all these experiments suggested one more thing is that the PKA, which is also acting like factor 11A, it's shown that PK is activating the feedback for producing more activated factor 12. But now we are seeing here that PK also activating downstream factor 9, like similar to factor 11A. But it could be maybe like you can see here, it's clear. But we use another factor deficient plasma, like factor 11 deficient plasma, which does not have any factor 11, but it has PK, and 3E8 can block PK binding to HK. So as you can see here, whatever effect we are seeing here by 3E8, that is definitely due to blocking the PK binding to HK, which will not allow its activation. And you can see clearly there is a very much significantly delay in the clotting, intrinsic clotting. So that signifies here that 3E8 blocks the PK binding, and PK is not getting enough activated. PK cannot get activated, and it blocks the further downstream coagulation. So our research highlights here that PK works like a factor 11A, and obviously when you add factor 11A, you will see the correction because that activates the downstream pathway. Next slide. So one more interesting thing we found here, which we want to highlight that sorry again for the title. So we found that it's well established that factor 10 in the downstream pathway, which is the common pathway, is required to directly cleave prothrombin to thrombin. And if you don't have factor 10, there are bleeding complications and also like hemorrhage and other conditions appear. So what we found here, when you use the factor 10 deficient plasma, which is definitely intrinsic clotting is delayed. But when we add 3E8, it was further delayed. And adding PKA, it did not correct it. So probably if you don't have factor 10 here, PKA, it's not able to show the effect downstream. But what I found here, like when we add the activated factor 11, which we confirmed that it was highly purified, it did not have any other contaminant or any other enzyme, significantly corrected the clotting delay which was induced by 3E8. We did so many experiments, I'm just showing few of them here, that then we thought that maybe because factor 11, it's known that it cannot directly activate prothrombin to thrombin. So maybe it's activating factor 9, which somehow acting like factor 10 and somehow involved in the cleaving prothrombin to thrombin, at least in the factor 10 deficient plasma. So when you use a factor 9 deficient plasma and perform the extrinsic clotting, as shown in the prothrombin time, the PT is not delayed because you already have a robust enzyme factor 10. But when we tested in the factor 10 deficient plasma, like using an antibody which blocks the factor 9 activation, we see that the plasma did not clot. That means that at least in the factor 10 deficient, factor 9 is involved in the downstream clotting, like cleaving prothrombin to thrombin. We still don't know how it is involved. We are doing more research on that, but just quantification here. I'm showing that the normal pool plasma, when you block the factor 9 and perform the extrinsic clotting, it had no effect because you already have factor 10, which is very robust enzyme for generating thrombin. But if you don't have factor 10 and you add anti-factor 9 antibody, which is specifically it's a monoclonal antibody, it will block and you don't see clotting, at least in this 20 or 30 minutes. So that means factor 10 deficient individuals, the factor 9 is also involved in the clotting. That is also our finding. Thank you very much.
So yeah, thank you both for this nice summary of your data. And for all which are joining us live, you can type your questions in the comment section. And to give you some time to do this, I will start with a few questions already. So one of my question, if PA can substitute for factor 11A, then why is factor 11A deficiency associated with bleeding conditions?
That's a great question, Timo. So we've clearly shown that PKA can substitute for factor 11A in our assays, but it's much less efficient than factor 11A itself. So we believe that's why there's still bleeding complications with factor 11A deficiency. It's also been shown that factor 11A can directly activate thrombin, but it's not known yet if PKA can do that. So it could be that if you're also missing the direct thrombin activation by not having factor 11A present, that's going to lead to more severe bleeding complications.
Sorry, and just I want to add it here that thrombin can activate factor 11, so that's the feedback. But maybe even though PK and factor 11 has similar like structure and they are similar in kind of enzyme, but it's not known whether thrombin can activate PK to PKA. Yeah.
Okay, thanks a lot. And yeah, my next question, since your antibody is blocking the domain 6, I assume that there are at least 5 other domains, so which might have different functions. So so can you kind of comment if the antibody only blocks the effects like the binding of factor 11 and PK, or if there are also effects on the other domains of, like, for example, in terms of platelet aggregation, something like this.
Sure. So we know that just recently we did a study. We know that 3E8 does not interfere with platelet aggregation. And we also know that 3E8 does not interfere with HK's binding domain, which is domain 5. It does interfere with cleavage of HK, which is in domain 4, but this is just an indirect consequence because you don't get binding of PK, so you don't get activation of PKA, and then therefore you can't get cleavage of HK at domain 4. So it's an indirect interference with domain 4. Otherwise, we don't know of any other domain interaction. Pradeep, do you have more to add to that?
Yeah, I think that's how the antibody is designed to block the bradykinin generation. So PKA can only block, like cleave the domain 4, which produces the bradykinin, and then when you block it, so you don't get bradykinin generation. So it has two functions. Like one is like it's blocking the clotting things, and one is like blocking the bradykinin generation. So that's the mechanism.
Okay, thanks. And yeah, I think we don't have a question so far from the other listeners, so I would continue with another one. So factor 12 can also become activated independently of plasma kallikrein. And does your antibody affect this activation as well, or not?
So yeah, it did not affect that. We tested that. Like definitely, you're right that factor 12 can get auto activated on surface, but when we tested that, 3/8 antibody did not affect the autoactivation. It definitely affects the feedback activation of factor 12 through 3 kallikrein, but it does not affect autoactivation. And we did not see effect of 3-8 in the peak factor 12 deficient plasma.
Okay. And then, yeah, I mean, one obvious question, at least for me, who I'm also working with antibodies, is like, are there any therapeutical implications? For example, using your antibody, have you ever done some in vivo experiments? And can you just comment on this?
So, yeah, Erin, do you want to say about
Oh,
that?
sure. So our hope is that obviously we could use 3E8 therapeutically or even prophylactically, like for something such as hereditary angioedema. So we're in the process of taking this in vivo and working on some model systems. So hopefully we'll have more information on that soon.
And one more thing I want to add here, as we showed here that factor 9 can act like factor 10 in the factor 10 deficient individuals plasma, but what we see in the factor 10 deficient individual, like factor 9 may not be as strong as factor 10. That's why factor 10 deficient individual, they still have hemorrhage or very severe bleeding complications. Even the mice lacking factor 10, they die within a few weeks. So, what is our conclusion here? That maybe it's not very important. This pathway, the alternative pathway of factor 9 activating thrombin, may not be very important physiologically. Maybe it's contributing a little bit, but there are disease conditions like systematic amyloidosis, where factor 10 gets depleted from the circulation, and they are very prone for the hemorrhage. These patients are also prone for severe hemorrhage and death. And in many patients, it had been found that factor 9 is also less. Like factor 10 is less, factor 9 is less. So now we know that if we block factor 9 in factor 10 deficient plasma, the bleeding could be very fatal because it will not clot. So at least we can suggest, like in these patients like with the systemic amyloidosis where factor 10 deficiency and factor 9 deficiency both are present, they should be routinely tested for both the factors, or maybe this combined deficiency through our mechanism proposed here contributing to some of the hemorrhagic conditions and death in these patients.
Okay, yeah, thanks for this important note. And yeah, I think there are no questions in the chat. And yeah, then I would like to thank both of you again for the presentation and for answering all my questions. And yeah, for those who listen to us, please also check out the original publication. And I think if you're more interested into the topic, there's also a review by these two published at RPTH, which you can also check out. And then I also want to thank Luke, who worked in the background and organized everything and helped us with the technique. So that was very nice. And yeah, thanks again, everyone. Don't forget to follow us on social media and to read the papers. And yeah,
And
thanks a lot.
sorry about that figures are not showing in the presentation. Like I'm sorry for that.
No worries. I mean, you explained it well. And yeah.
Thank you very much,
Thank
Timo,
you
and
very
thank you
much.
to
And thank
RPT.
you all the listeners. Yeah.